Swayam Prabha. Digital India. Educated India.
Today we will be starting off with the second part of innate immunity, and so before we go into that, we would it might be a good idea to summarize some of the main aspects of the last lecture. The first is the importance of innate versus adaptive. As mentioned previously, innate is a quick response. It is non-specific. It does not differentiate between different types of bacteria, but it tells the host that there is some sort of a invasion that has taken place, and that is the most important thing. It is an evolutionary conserved process, and it is present in lower organisms such as Drosophila, horseshoe crabs, etc. So, it would be a good idea for you to sort of think about where the Drosophila and horseshoe crab were useful in the previous lecture. For example, the use of the identification of toll receptors was shown in Drosophila, and the measurement of lipopolysaccharide, which is a potent endotoxin, is used using lysates from the horseshoe crab. We also talked about physical barriers that are important in innate immunity, for example, epithelial cells, mucus. So, mucus that is produced traps these microbes, and that is useful in sort of containing their spread. There was also discussion on different types of cells, for example, neutrophils, macrophages, NK cells and dendritic cells. So, we will briefly go a little bit over them. Neutrophils are one of the first cells that the host responds to, and they are the first ones to arrive at the site of pathogen entry. Subsequently, they produce chemotactic factors, and macrophages are recruited. Macrophages are important in processing and presenting antigens to T cells. Then you have natural killer cells, which are important for what is known as antibody dependent cytotoxicity. Once antibodies are produced, these target cells. They are also important for tumors, and dendritic cells are perhaps the physiologically most important antigen presenting cells, which present different antigens to T cells, and so you can turn on the adaptive immune response. We had discussed about the fact that the innate immunity modulates adaptive responses, and this is mainly seen in the use of microbial adjuvants, microbial components in adjuvants. So, for example, complete Freund's adjuvant contains killed mycobacteria, and this was known as the immunologist's dark secret by Charlie Janeway, who first propounded that the innate components needed to be activated to get an optimal immune adaptive response. And subsequent studies resulted in identification of toll-like receptors that are present in host, which recognizes specific microbial components. So, TLRs, for example, they contain this external leucine rich repeats, and these LRRs or leucine rich repeats are important for protein-protein interactions, and it contains an internal IL-1 receptor signaling domain by which the signal transduction can be done. We also discussed the ways by which LPS is detected and the response by host. So, LPS binds to LPS binding protein in the serum, and this complex is transported. It is recognized by CD14, and TLR4 is important for recognition of LPS, and this complex alone cannot signal. It needs a signaling molecule known as MD2. Which is important in signal transduction. So, you turn on the cytokine response, and you turn on an acute innate response, which is often manifested in with respect to cytokine release, increased phagocytosis, killing of target cells or pathogens, etc. We also discussed an important part, which is septic shock, and septic shock is quite prevalent, especially in hospitals post infections, and this is important because you have an acute inflammatory reaction because of the presence of microbes, and the host response is so strong that it often leads to multi-organ failure, low blood pressure, and sometimes even death. So, it is important that these aspects are revised by you before we move on to the next part. Okay. So, we had mentioned the role about signaling pattern recognition receptors, and the ones that we mainly discussed in the last lecture were toll-like receptors. Now, there are two main types, there are others too, but the two main signaling pattern recognizing receptors are the TLRs and the NODs. So, what are the NODs? The NODs are the nucleotide oligomerizing domain. These are intracellular sensors. If you remember, most of the TLRs, TLR2, TLR4, etc., they are present on the surface. Some TLRs are present in endosomes, for example, TLR9, but in case of NODs, they are primarily intracellular sensors. So, they sense microbial entry pathogens within cells. And these are characterized by particular domains. So, NOD proteins contain an N-terminal domain, which contains the CASP domain or the caspase recruitment domain, which is important in apoptosis and activation of NF-kappa B. We will discuss this important transcription factor NF-kappa B subsequently. The middle domain contains the nucleotide binding oligomerizing domain, and hence the name NOD, which is important in self oligomerization. The C-terminal domain contains the leucine rich repeat, which is important for protein-protein interactions. You will remember that DLRs contain LRR domain, but it is present on the external surface, whereas in NODs it is present in the C-terminal end. The importance of NOD has been shown with its association with Crohn's disease, which is an inflammatory bowel disease. Now, mutations in NOD2 lead to have been associated with Crohn's disease, which is an excessive activation of macrophages and T cells in the bowel. One of the possible reasons for this is that has been shown that NOD regulates TLR2 signaling. So, for example, in TLR2 knockout mice or mice that lack TLR lack NOD, they secrete large amounts of IL-12 in response to TLR activation. So, it is possible that NOD is playing a regulator of the TLR activation, and since the mutations in NOD are unable to control this, it results in greatly exaggerated activation, possibly resulting in inflammatory bowel disease. There is also a reduction in defensin production, which are important antimicrobial peptides in NOD2 knockout mice, and that may also be important because as previously mentioned, that the production of antimicrobial peptides is important in reducing the number of bacteria that is present in the intestine. Okay. There are other types of pattern recognition receptors, and two of the important ones are endocytic. Now, so these do not signal by themselves, they are endocytic, which means they will bind to complexes, and these receptors internalize and clear off these bound ligands. So, one of the important ones is scavenger receptors. Scavenger receptors are particularly important because they bind to modified lipoproteins, for example, LDL, which is important in transport of cholesterol. So, they also bind to charged polyanionic ligands, bacteria, apoptotic corpses, etc. And this part is important because, for example, when there is increased cell death, you want to remove of the dead cells, and scavenger receptors may be playing an important role in these sort of processes. There are also mannose receptors that are present on macrophages, and these recognize high mannose containing proteins which are present on surface of microbes and which are then ingested by the macrophages. Okay. We will next be moving on to the complement system, and the complement system has several functions. It is important in the control of inflammation. It is most important in the clearance of immune complexes. So, especially when you have antigen antibody complexes, they need to be cleared off, and the complement system comes in place over here. They are important in activation of the antimicrobial defense, and we will see parts of that subsequently, and it is a major effector of immunopathological diseases. So there are different ways by which complement can be activated. The most classical and well studied is the antigen bound to antibody. So you have antigen bound to antibody, and this activates complement. Okay, and this is useful in the body. It's also useful for doing in vitro experiments where you want to isolate certain populations of cells. You have an antibody to a particular cell type, and you can use complement to deplete that particular cell type. So complement has a variety of uses. But in terms of innate immunity, one of the ways that it plays an important role is through the alternate pathway, where you have the activation of complement protein C3b, which binds to certain microbial surfaces and then gets activated and remains activated, and as a result of which the cascade initiates. The other way by which complement can be activated is through the mannose binding lectin, which binds to pathogen surfaces, and we will discuss these in slightly greater detail. The important part of the complement pathway is that the proteins in this system act as an enzyme cascade. So one protein gets activated, in turn activates the other one, and so on until the microbe is lysed, and we will see that somewhat later. So here what is shown over here is the classical pathway. So here you have antibodies, and these antibodies have been produced against the microbes, and these results in what is shown over here as antigen antibody complexes, and these antigen antibody complexes are clumped together, and then complement binds to these, and what it does is it results in lysis of the microbes, and as a result of which these antigen antibody complexes and the microbes are lysed. And what is shown over here is the cascade, as I was talking about. So C3b is a opsonin, which means it enhances the phagocytosis by coating. So once C3b is coated on microbes, it enhances phagocytosis. That's the process of opsonization. It also results in activation of the complement, and you can see the cascade leading to the microbial plasma membrane loss or lysis of microbes. There are other processes involved in here. Complement plays an important role in inflammation. So it increases the blood vessel permeability and the chemotactic attraction of during phagocytosis. An important disease that is related to this particular pathway is the common opsonic defect and its relationship with mannose binding lectin, something that we had just discussed in the previous slide. The mannose binding lectin is a host protein. It binds directly to mannose N-acetyl glucosamine, that is Foucault's residues, etc., that are present on microbes, and directly activates complement. So this is the third pathway by which complement can be activated. What is important is that deficiency in MBP leads to common opsonic defect, that is an inability to phagocytose microbes by neutrophils, and this defect affects 5 to 6% of individuals, which is fairly high, and it is commonly detected in children with recurrent infections. So how was it discovered? What was found is that neutrophils from patients who lack MBL or have mutations in MBL were unable to phagocytose yeast, which is Saccharomyces cerevisiae, but the defect was reversed when serum from healthy donors was used in the same assay. So there was something in the serum that was missing, and subsequently it was identified to be mannose binding lectin. Okay. Complement receptors are important over here. The most perhaps the most important one is CR1, which is present on erythrocytes, or SCR stands for complement receptor, and it is mainly responsible for clearance of opsonized immune complexes. CR1 amounts decrease in aged RBCs. So as RBCs get old, these amounts decrease, and they are also low in diseases involving clearance of antigen antibody complexes, for example systemic lupus erythematosus, and that is again something that will be discussed in the lecture on autoimmunity. CR2, the complement receptor 2 is present on B cells, and it allows for enhanced response to antigen. So you can imagine a situation with B cells, and if you have the antigen, the CR2 complement and bound to antigen antibody, and it's binding to B cells, so it's internalized efficiently, and this allows for better presentation and activation of B cells. So, so CR2 is important for enhanced B cell responses. CR3, which is shown over here as CD11B and CD18 present on neutrophils, NK cells and macrophages, and it's important for phagocytosis and destruction of foreign cells. We will see the importance of this particular subunit CD18 subsequently. Okay. The other molecules that are important in adhesion in inflammation is adhesion. During the process of inflammation, adhesion receptors increase. So, both receptors as well as ligands increase, and this adhesion is important especially because microbes have to or neutrophils, macrophages have to travel to different have to leave the blood circulatory system and travel into tissues where damage has taken place. So, in order to do that, adhesion plays an important role by which they can go to particular areas within the part where tissues are affected, and adhesion receptors and ligands play a very important role in this process. So, one important disease is known as leukocyte adhesion deficiency, and this results due to mutations in CD18 and CD18 is a common beta subunit. Now, this beta subunit, it's associated with different types of receptors. For example, CD18 is important in CR3, which is the complement receptor 3. Here CD18 is associating with CD11B. Alternatively, CD18 is also important for LFA, which is important which is an important adhesion receptor. Here the alpha subunit is different. It is CD11A and which associates with CD18. So, you can see here that CD18 is common in both these two different types of receptors, but the alpha subunit is different. If you have patients that have mutations in CD18, what it does, it affects proper cell surface expression and function of both complement receptor 3 and LFA molecules. Okay. What happens as a result of this is it results in defects in adhesion. So, as a result of which the cells will not be able to migrate to the affected area, as a result of which diapedesis, which is this ability to migrate, is affected. Consequently, patients suffer from recurrent skin infection, pneumonia, septicemia, gingivitis, which is inflammation of the gum, impaired wound healing, etc. So, it shows you clearly, these examples are there to show you the importance of particular subunits in the innate immune response. Okay. There are other molecules that are important. So, FC receptors will bind to antigen antibody complexes, and these signal. FC receptors are particularly important for example, in signaling during allergies. Then you have chemokine receptors, which are required for trafficking to different tissues or sites of inflammation. An important chemokine receptor is CCR5, which is important for entry of HIV. In the last class, we had talked about CD5 positive B cells present in the peritoneum, or Li1 B cells in the mouse as they are known, and these are often responsible for production of what is known as natural antibodies. So, these antibodies are produced in response to different types of microbial pathogens, so on. So, it is naturally present, and mice lacking natural antibodies are 10 to 100-fold more sensitive than their wild type compartments in resisting infection by microbes. So, it clearly shows you that natural antibodies are also playing an important role in innate immunity. An important class of proteins that play a response in innate immunity are accused phase response proteins. These proteins are produced rapidly in response to inflammation, and the liver is responsible for production of several acute phase proteins. An important acute phase protein is the C-reactive protein, and it binds to phosphocholine present on dead or dying cells and some bacteria in order to activate complement. So, as a consequence of that, it binds to apoptotic cells in a calcium dependent manner. So, CRP is important for of dead cells, and it is a way of which you are once the inflammatory reaction is over, you are sort of down modulating and getting rid of all the debris that is around. It is a way the body has developed by which dead dying tissue can be removed efficiently. Among the soluble factors that are produced during inflammation are cytokines and chemokines. Some of the important cytokines are IL-1, IL-6, IL-12, TNF. And in fact, TNF is a marker because it is so rapidly, it is one of the quickest or the fastest produced cytokines during inflammation. So, it is often thought to be a marker for inflammation. Now, what happens often during signaling via the pattern recognition receptors, one of the consequences, one of the downstream consequences is our production of cytokines, which have a variety of effects. Also important is the production of chemokines, and chemokines, especially the example shown is that of IL-8, which is important in attracting neutrophils during infection. So, the production of IL-8 attracts neutrophils to the site of infection. Okay. So, this slide depicts the main types of chemokines. You have the CC type shown by MCP rantis and the CXC shown by IL-8, which is a neutrophil attractant, and as mentioned to you that CCR5 is important in playing an important role in HIV infections. So, chemokines have several different roles, and at this point we will not dwell on this further than to show you that they play a variety of roles. In cytokines, and important cytokines are known as interferons. Interferons, it originates from the word interfere, and interfere because interferons were discovered to interfere with viral replication. So, in terms of antiviral immunity, the interferons are known to play an important role. There are two main types of interferons, type 1, which is IFN alpha beta, IFN alpha, IFN beta, or type 2, which is interferon gamma. And the type 1 interferon, which is interferon alpha beta, is involved primarily in antiviral immunity. It has other functions, but its main role is, it's very well known to play an antiviral role. So, how does the type 1 interferons function? There are several mechanisms that are known. One of the important mechanisms is via the production of MX GTPases. What these GTPases do is they inhibit transcription in one case, but more importantly they inhibit viral assembly. So, they inhibit with the transport, they interfere with the transport of viral capsids. They also sort them to locations where they are not available for assembly. So, the GTPases prevent or slow down a viral assembly and viral assembly. The other main way by which is through the production of this enzyme known as 2,5-oligoadenylate synthase. What this does, it binds to double stranded RNA and forms these 2,5-oligoadenylates. So, they adenylate these adenylation and as a result of which this in turn activates RNase L, and which degrades single stranded RNA, and as you will remember that these are important during production of viruses and often also for replication of viruses. So, this is and transcription of viruses. So, this is one way by which it acts in an antiviral manner. The other way is there is phosphorylation of eukaryotic initiation factor 2, as a result of which translation is inhibited and viral proteins are not efficiently made. Apart from their strict antiviral roles, type 1 interferons are now shown to be important in other processes in modulating host immunity, one of which is in maturation of dendritic cells, generation of cytotoxic T lymphocytes, and in some cases they have been shown to increase the survival of T cells. Type 1 interferons are of use clinically. For example, type 1 or interferon alpha along with the antiviral drug ribavirin is used to treat liver diseases with chronic hepatitis B infections. The second case is interferon beta. It is used to treat multiple sclerosis. Now, how does it do in this particular case, interferon beta is anti-inflammatory and reduces T cell migration to affected neurological tissues. Also, it increases the production of anti-inflammatory cytokines. The other interferon is the type 2 interferon, interferon gamma. Now, interferon alpha beta is produced by all different types of cells, whereas interferon gamma is produced mainly by T cells and natural killer cells. And what interferon gamma does is it activates macrophages, and the way interferon gamma functions, it is primarily through induction of expression of several immune genes. For example, transporter associated with antigen processing, C2TA, which is important, which is an important transcription factor for MHC class 2, NOS2, GP91-FOX. The NOS2 is important in the production of nitric oxide, GP91-FOX is important in production of superoxide radicals, and these two are ones that we will see a little bit later. Interferon gamma is a potent inducer of MHC class 1 and MHC class 2 expression. So, once the MHC molecules are increased, the chances of peptides that derive from pathogens are also increased, because overall the production of MHC molecules increase. This is especially important during inflammatory conditions during infections. It is a key cytokine in resisting microbial infections, and it modulates a T cell T helper differentiation. Perhaps the most important role of interferon gamma is seen in patients where inoculation of BCG, which is a live vaccine given to prevent tuberculosis, and if this live vaccine is given to children that lack interferon gamma, interferon or its receptor, IL-12 or IL-12 receptor, it results in bacteremia known as BCGosis. So, one of the primary roles interferon gamma is in boosting up of immunity against intracellular pathogens. Okay. So, there are other types of molecules, and we will briefly mention or go over the different other types of molecules, one of which are the collectins, and these are calcium dependent lectins. What do we mean by lectins? These are sugar binding proteins that recognize pathogen associated molecular patterns, okay. And these are important because they are involved in direct opsonization. If you remember, is the process by which there is enhanced phagocytosis of opsonized bacteria or microbes, neutralization, agglutination, complement activation, and phagocytosis to curb microbial growth. There are different collectin members, and if you remember, the mannose binding lectin is an important it's an acute phase protein. That means it is produced rapidly during the inflammation, and it is also important for complement activation. Is a member of the collectin family. Two of the members of the collectin family, namely surfactant protein A and surfactant protein D, are well characterized to play an important host response in the lung. So, these are ways by which these are different proteins that are produced by the host in order to be able to tackle different types of microbes, because we are constantly under attack. We had discussed antimicrobial peptides in the previous class, and we will again over some important aspects of it. These represent one of the first line of defense in epithelial surfaces, and this is especially true in the intestinal or the gut lumen. You will also remember that TLR and the IMD pathway activation in Drosophila results in production of antimicrobial peptides. For example, drosomycin, which is antifungal, and drosin and dipterin, which is antibacterial. TLR activation is thought to increase cytokine production in others in mammals, but in case of Drosophila, the main role has been shown to be in production of antimicrobial peptides. Okay. So, there are different families of antimicrobial peptides. You have defensins, which are small cationic antimicrobial peptides. And defensins are produced by neutrophils, macrophages, and Paneth cells. Paneth cells are present in the intestine, and they are potent sources of defensins. Catalysacids, they are produced as large precursors, and then they are trimmed to produce these antimicrobial peptides, and they are found on the surface of gastric intestinal cells. The other one is lysozyme, and lysozyme, as you should know, it is the first enzyme and the second protein to be crystallized. It hydrolyzes N-acetyl glucosamine and N-acetyl muramic acid bond, which is present in several bacteria. Lysozyme is present in our tear secretions and other fluids, and it helps in in cleavage of bacteria or lysing of bacteria in a non-specific manner. So, there have been once people started working on defensins, what was shown is that these are small peptides and they are able to insert themselves into microbial membranes and cause their lysis. What has been shown is that their direct antimicrobial action was well known. What people are beginning to appreciate now is that defensins also play a role in host immunity. So, for example, in antiviral defense, they act both on the virus as well as on the host cells. They have been shown to have chemotactic activity for T cells, monocytes, immature dendritic cells, and they induce cytokine production by monocytes and epithelial cells. Okay. So, this is an example to show you the role of human beta defensins during rhinovirus infection. So, over here you have rhinovirus infection. This is it is affecting this mucosal epithelial cells, and this is produced in response to double stranded RNA intermediate that is present. And if you have poly-IC, which is used as an artificial instead of the double strand intermediate, you use poly-IC and activation via TLR3, that also results in production of the human beta defensin 2 and 3. Now, so that is essentially that was shown. Whatever I said over there is written over here, but in case of another viral infection, in this case HIV, the production of human beta defensin 2 and 3 does not require viral replication. So, you have situations in which in some viruses you need viral replication for production of these defensins, in other viruses you do not need this, but nevertheless they play an important role in antiviral immunity. Okay.
I will just change the battery.
Sorry.
Maybe
okay.
It may go and
okay.
That's fine.
All right.
It's precautionary.
Part of
yeah, from the previous sentence or something like that. No problem.
we can stop in the middle also. Okay, that's not a problem.
So I think you're feeling nervous when you get a picture in picture. It's okay.
This is better
Okay.
for me. If I see myself, I get nervous.
What I'll do, sir, I'll make you in the corner. We'll make little bigger picture.
That's fine. I mean, you know what you have over there is fine.
Thank you. Okay, fine. I think you can start.
All right. Okay, so as I was saying, so the production of human beta defensin 2 and 3 in some cases requires the production of an RNA intermediate, and in some cases and viral replication, and whereas in other cases it does not require. Nevertheless, the defensins play an important role in antimicrobial immunity, and this was an example to show you a role of these. This is a lysozyme. Again, it's highly active against gram-positive species, mainly because of the part that it cleaves the muramyl peptide bond is present primarily in gram-positive. And there are other means also. So, they activate bacterial autolysins, they result in bacterial aggregation and so on, and so they play important antimicrobial roles. The other ones are the catalycidins. Remember, the catalycidins are produced as larger precursors and they get trimmed down over here, and catalycidins play an important role in the intestinal lumen, and they are present on the surface of gastric intestinal cells. Lactoferrin is a globular glycoprotein, and it is present in secretions of the saliva, tears, etc. It is present in the highest amounts in colostrum. So, why is colostrum so important? Colostrum is important because it is the first milk that babies drink after birth, and it is possible that lactoferrin is playing an important antimicrobial role, and because it is helping the baby in fighting immunity, because once the babies are born, they are more susceptible, they do not have their own immune system of their own, and they rely a lot in the mother. Their immune response from their mothers. So, perhaps for the initial few months, lactoferrin may be playing an important role because it is present in very high amounts in colostrum, which is the first milk that is produced after birth. Lactoferrin is also released from neutrophils and respiratory tract epithelium. It has multiple roles. It is anti-inflammatory, antiviral, anti-LPS, anti-biofilm. And biofilm is important because of what happens in some cases, bacteria form from these film-like structures. So, they form a sort of a colony of their own in different tissues, and these biofilms are highly resistant to antimicrobial drugs. So, lactoferrin has this anti-biofilm property, which is very useful. This is a list of different viruses that are susceptible to lactoferrin. And this slide summarizes the different roles of lactoferrin. So, basically it is a fungicidal, which means, like, for example, it is shown as against Candida. It has antiviral roles, HIV CMV is shown over here. It has anti-inflammatory because it is anti-LPS. It is also important for bacterial killing, and most importantly it has anti-biofilm properties which make it important, because you can see that this is a film, a film like structure that the microbes are sort of developing, which makes them very resistant to treatment with antibiotics, so on. So, the anti-biofilm property of lactoferrin is very useful. Okay. During the studies on the signal transduction of TLRs, we had shown importantly the activation of NF-kappa B. This NF-kappa B and the NF-kappa B equivalent in Drosophila is, the cascade is, the signal transduction cascade is almost similar. So, for TLR, for toll activation and toll-like receptor activation in mammals, NF-kappa B is playing an important role. So, you can see the conservation of both the receptor, the signal transduction, and especially an important transcription factor like NF-kappa B, and it tells you about the conserved signaling and processes between in innate innate immunity in lower organisms as well as higher organisms such as mammals. So, NF-kappa B was first described as a nuclear factor a long time back. At that time it was shown to be important in transcription of the immunoglobulin kappa chain in B cells, and hence the name NF-kappa B. Subsequently it has been shown to be important in production of wide variety of molecules, especially those related to inflammation. So, for example, in production of cytokines, acute phase proteins, adhesion proteins, NF-kappa B plays an important role. What is shown over here is the signal transduction pathway. So, NF-kappa B is usually present in the cytosol, and it is associated with another protein known as inhibitor kappa B, which is shown over here. So, you have NF-kappa B and I-kappa B in this complex. Upon signaling, what happens is that there is ubiquitination of inhibitor kappa B and degradation of the inhibitor. So, as a result of which, and this degradation is via the 26S proteasomal pathway, and you have over here what is shown as the activated NF-kappa B. Now, once NF-kappa B is activated, it can now go from the cytosol into the nucleus, where you can bind to its particular cognate binding sites in front of promoters and turn off transcription of several genes. And what is shown over here is apoptotic factors, cytokines, cell cycle regulators, so on and so forth. So, this pathway of activation of NF-kappa B is important, and it is very important for students to understand this particular pathway. Remember, the NF-kappa B activation pathway is again conserved between Drosophila and mammals, and the signals are also conserved for this activation, which is the toll and the IMD pathway, resulting in activation of the Drosophila NF-kappa B, whereas in mammals it is the TLR and activation of the NF-kappa B pathway, and it results in a wide variety of responses, a very important concept for students. So, this is shown over here, NF-kappa B, it results in transcriptional activation, and you have synthesis are now of several immune related molecules. You have reactive oxygen intermediates, antimicrobial peptides, cytokines, chemokines, adhesion molecules, acute phase proteins, and so on. This is just a partial list, but the NF-kappa B is a super transcription factor. It turns on several different molecules. One of the ones that was shown over there was the induction of reactive oxygen species, and what I will do here is to discuss the importance of reactive oxygen and nitrogen species in innate immunity. Over here, ROI and RNI play a very important role in innate immunity, and the cells that are mainly responsible for these are the neutrophils, which rely mainly on the reactive oxygen intermediates, and macrophages, which rely mainly on the reactive nitrogen intermediates. However, the two pathways can converge together to form a potent oxidant, which is peroxynitrite, and I will go over this a little bit slowly. So, here you have oxygen, which is converted by NADPH oxidase. Now, NADPH oxidase needs to be assembled in the membrane upon activation. Once it is assembled, it can form superoxide, and the superoxide, which is highly potent, it can be converted into hydrogen peroxide, or it can combine together with nitric oxide to form peroxynitrite, which is a potent oxidant. And then these are the hydrogen peroxides can go on to form hydroxyl radicals, and these will be a part of the reactive oxygen intermediates. The other one are the reactive nitrogen intermediates, and here for the production of nitric oxide, arginine is the donor, is the nitrogen donor. It combines over here, and you have nitric oxide synthase, which is responsible for the production of nitric oxide. Now, in the body there are three main types of nitric oxide synthase. You have the eNOS, which is the endothelial NOS, the neuronal NOS, nNOS, and then you have the inducible NOS. For the purpose of this class, and especially with respect to the immune system, what we are referring to is the inducible or immunological one, or the iNOS, or commonly known as NOS2, which is shown over here. Now, what happens with nitric oxide, it can combine, as shown over here, with superoxide to form peroxynitrite, which is extremely potent oxidant. But nitric oxide on its own has a lot of roles too. The main one that was initially shown was activation of guanylate cyclase, resulting in the production of a cyclic GMP, and this cyclic GMP will have its own roles. The activation of guanylate cyclase is because of nitrosylation of heme, which is present in the active site of this particular enzyme. The other way by which nitric oxide functions is by an S-nitrosylation of proteins. So, SNO, which is a cysteine, and then SNO. So, that is how it nitrosylates different proteins, for example, hemoglobin, glutathione, RAS signaling proteins, so on and so forth. So, this is an important way by which the reactive nitrogen intermediates and the reactive oxygen intermediates play an important role. And remember, these are potent molecules, and they would kill different microbes that are ingested, and often the cells also themselves get killed, and which are then subsequently cleared off by the process of phagocytosis. So, the cells sort of not only kill microbes, but they might get themselves killed because of production of these radicals, and then ultimately the body is saved because of the fact that the microbes are killed, and then the debris and all are cleared off later. Okay. Important molecules in the inflammatory process are prostaglandins, and they are derived from fatty acids. Arachidonic acid is an important one, and they play important physiological roles, for example, in pain, fever, inflammation, so on. The enzyme that is responsible for production of prostaglandins are cyclooxygenase. You have COX-1 and COX-2, and which are present in blood vessels, stomach, kidneys. COX-1 is responsible for the basal production of prostaglandins, and the increased production due to stimulation is done by COX-2. The receptors for prostaglandins are cell surface G-coupled receptors. Now, the importance of prostaglandins comes into focus by the use of non-steroidal anti-inflammatory drugs, and which several of us take whenever we have headaches, pains. We take aspirin, ibuprofen, naproxen, so on. These belong to the category of NSAIDs. So, and the way it functions is that they are inhibitors of COX. Now, aspirin is particularly important because not only is it an inhibitor of COX-1, it also reduces platelet aggregation as shown, and these reduces adverse cardiovascular events. And for heart patients, often the doctors prescribe aspirin, and now you know what is the mechanism of action, because it reduces platelet aggregation and and reduces the risk of adverse cardiovascular events. So, aspirin has two roles. One is a COX-1 inhibitor and also reduction of platelet aggregation. Now, once you have the inflammatory process, anything that goes up needs to come down, because remember, if the activation keeps on in a sustained manner, it results in problems to the cells and to the host. Sepsis is a good example of that, where you have so much activation of immune cells that it becomes very difficult to control the process, and ultimately it leads to multi-organ failure and death. So, in the immune response, whatever goes up, if the immune response is induced, it needs to have some regulatory mechanisms by which it can be reduced. And this brings us to an important aspect, which is down modulation of inflammation or down modulation of the inflammatory response. So, with inflammation, anti-inflammatory processes are also induced. For example, anti-inflammatory cytokines, you have TGF beta, IL-1, IL-4, IL-10, these are well known anti-inflammatory cytokines. Some hormones also play an important role, for example, glucocorticoids, and these are produced by the adrenal glands, and they are anti-inflammatory. They are useful in case of allergies, asthma, and so on. In case of heightened immune reactions, as observed in case of autoimmune diseases, sometimes glucocorticoids are also prescribed. The other way, once you have debris, they need to be cleared off, is apoptosis, and so macrophages are often responsible for recognizing apoptotic cells because of the expression of eat me flags. So, these cells that are undergoing apoptosis, they express certain cell surface receptors. These are recognized, and then they are phagocytosed by macrophages. One of the eat me flags is phosphatidylserine, which is usually present inside, but in apoptotic cells it is present on the cell surface, and this can be recognized using annexin 5, which binds to phosphatidylserine. Now, this particular assay is useful in detection of apoptotic cells. So, as if you wish to look at cells undergoing apoptosis, often you can look at the surface expression of annexin 5, which is a marker for apoptotic cells. Now, in terms of TLR induced responses, again there are different mechanisms in place to reduce TLR activation, because microbial infection is often associated with TLR responses. So, initially I had shown you the role of NOD2 in regulation of TLR responses, and it is possible that NOD2 mutations, and may explain as to how NOD2 mutations lead to Crohn's disease. Now, another molecule shown, TOLIP. Now, TOLIP was shown up in my previous classes lecture slides on TLR, but I did not explain it because there was not sufficient time to explain each and every molecule. But TOLIP inhibits IRK, and this IRK activity is associated with TLRs. So, again, so you have IRK activities associated with TLRs, you have a regulator of this particular activity, so you can reduce TLR activation. Non-pathogenic bacteria also reduce inflammation, and in fact they block inflammation induced by pathogenic bacteria. You will remember that in our gut we have lots of non-pathogenic bacteria, and there are different mechanisms in the gut to take care of these huge load of bacteria that are living in the gut. The peroxisome proliferator activated receptor, PPRA gamma. Now, peroxisomal proliferator activated receptor, these are nuclear transcription factors which get activated, and they lead to wide variety of responses. In this particular case, peroxisome proliferator activated, it results in increased production of peroxisomes, and that is why the name peroxisome proliferator activated receptor. And these might reduce inflammation, these have some role in inflammation, and by increasing nuclear export of the Rayleigh subunits of NF kappa B. So, NF kappa B goes into the nucleus once it is activated, and what this could be doing is that it is exporting this NF kappa B back into the cytosol, and by increasing the export of this, you are reducing activation. So, there are different mechanisms in place by which down modulation of responses can occur. The other example is shown by the A20 ubiquitin ligase, which down modulates TLR dependent responses. The other molecule which is well studied is SOX1, which is a suppressor of cytokine signaling. It is an inhibitor of the JAK-STAT pathway, which is important in signaling via the interferons. Okay, one of the examples of down modulation of inflammation is the example that is shown over here is the expression of SIRP alpha, which is an inhibitor tyrosine motif containing receptor on macrophages. Now, usually binding of this to CD47 results in down modulation of phagocytosis. However, as RBCs age, they down regulate CD45, and these aged RBCs are rapidly phagocytosed by macrophages. So, this is an example again of phagocytosis, about molecules that play an important role in phagocytosis and how you can down modulate the responses. I will briefly now talk about the relationship between the innate and the adaptive immune system. The NK cells produce interferon gamma. These activate macrophages, and once macrophages get activated, they will present and process more efficiently to T cells, and consequently they will modify your T cell responses, which can maybe of Th1, Th2, and so you can see a role where NK cells, which are innate, which play a role in innate immunity, are also modifying the adaptive immune response. The second one is dendritic cells. Some dendritic cells produce large amounts of type 1 interferons in response to viral infections, and these have been shown to play not only an important role in reducing viral replication, but they also result in increased numbers of viral specific T cells. So, again it is a case by which a classical innate cell is modulating the adaptive response. A fine example of immunity or this interrelationship between adaptive and innate is seen in the gut, because in the gut you have huge numbers of bacteria in there, and the gut plays an important role because you have this gut associated lymphoid tissue, and the upper bowel has lesser number of bacteria, but it reacts to dietary antigens, whereas the lower ones, in addition to the dietary antigens, you have huge commensal organisms living there. Over here you have in the intestine you have specialized patches of organized lymphoid tissue known as the Peyer's patches, and among the important cells which present uptake which is responsible for uptake of antigens and then giving it to the dendritic cells and the other cells in the lymphoid tissue are known as M cells, and these are epithelial cells, but which play an important role in this process. The IgA that is present in large numbers in the gut lumen is also an important role. In fact, mice that cannot produce IgA have much higher numbers of gut bacteria. So, the gut immunity or the gut process that is very important because of this fine interaction between innate and adaptive arms. Okay, I will also briefly discuss how plants and innate immunity function. So, what about plants? We have been discussing animals so far. So, before ending, I will just briefly mention the different mechanisms by which plants also have it. So, plants plants are also susceptible to viruses, bacterial pathogens, so on. So, how do they deal with this? So, first is by the production of antimicrobial products, for example, phytoalexins, salicylates, antimicrobial peptides, enzymes, chitinases. So, they also can shut down transcription, and they do so by the dicer pathway. So, it will turn transcription down. Then you have what is known as the resistant loci or the R loci, which allows recognition of specific effectors. Several of the R loci proteins contain LRR, which is the leucine rich repeat domains in their proteins, and which is an important mechanism. So, you can see that the R factors and the LRRs and the relationship, there is some commonality that one can see over here. An important response by which plants take care is through the production of what is known as the hypersensitive response, which is upon infection the cells around that particular infected tissue, they die off. And how they die off is not clear, but it is possible that plants are known to contain caspase related proteases, and whether these are playing an important role needs further studies, but the hypersensitive response is clearly an important way by which plants handle the infection by pathogens. I will now briefly summarize and just go over some of the main aspects that we have covered today. First is that what we have tried to do is study different aspects of the innate response. You have the complement activation. We have looked at complement activation. There are different pathways. There are 3 different pathways: the classical, the alternate, and the lectin pathway by which they can be activated. And you have receptors that are involved. You have the TLRs, you have the nodes, you have the complement receptors, you have the adhesion receptors. We also studied the important roles of interferons, especially the type 1, type 2 interferons, IL-8, which is an important chemokine, which is an important neutrophil attractant, CCR5, which is an important for HIV infections. Effectors, the reactive oxygen intermediates, the reactive nitrogen intermediates. Here FOX NADPH oxidase is important for it. Reactive nitrogen intermediates, NOS2 is the key enzyme that is responsible for production of nitric oxide. We have also looked at down modulation of innate responses, the production of anti-inflammatory cytokines like TGF beta, IL-10, IL-4, and you have further ways by which the production of glucocorticoids, and so that you can suppress immunity. You have different proteins, for example, that will suppress immunity, for example the A20 ubiquitin ligase, the TOLIP, which is an inhibitor, or SOX1, which is an inhibitor of the JAK-STAT signaling pathway, which is important by which interferon signal. We also studied the link between innate and adaptive immunity, especially the role of NK cells in producing interferon gamma and modulating T cell responses. Dendritic cells, some dendritic cells produce high amounts of type 1 interferons, and which in turn affects the antiviral activity and the production of antiviral T cells. Now, most importantly, I would like to conclude with a study of 4 diseases and a sort of a review of these. So, NOD2 is an intracellular sensor. It has been shown to play a role in, or it is associated with Crohn's disease, which is an inflammatory bowel disease. So, mutations in NOD2 result in increased inflammation of the bowel, and it is possible that NOD2 is playing an important regulator of the T cell responses. CD18 is the common beta subunit. It is important for adhesion, leukocyte adhesion deficiency. Then you have mannose binding lectin, common opsonic defect, which is quite common, and it is important in opsonization and complement activation. FOX, which is NADPH oxidase, and the production and its association with chronic granulomatous disease. So, if you do not have NADPH oxidase, or your mutations in NADPH oxidase which are not functional, you have chronic granulomatous disease, which results in recurrent bacterial infections. Overall, what these 2 lectures, innate immunity, have shed light on the cells, the mechanisms by which our innate cells play an important role. So, they are not only act as the border security force, but they are important in modulating the adaptive immune response. Thank you.